Okay, everyone. So, hello. I hope that you all are very much fine, and I am back with a special class with you on the chapter photosynthesis. So, there was so much of chaos going on around NEET 2024 syllabus, right? Whether to study what to study, what not to study. So, we all have made a proper syllabus for you. You can go and watch the shorts which is recently uploaded in this particular channel, screenshot those things and only study those things. There are few chapters which in which some of the things are added, like in molecular basis of inheritance, protein biosynthesis is added. All those things I have made it for you and have uploaded in that particular short. So you just screenshot it and study those things only. It's not required to study everything now. Just whatever NMC is saying as of now, study that only. If later there is any updates regarding the syllabus, there are some confusions in the syllabus, like some part which is given in which is not given in new NCERT, and they have asked to learn that part also. So I think so there can be a major update in the in the syllabus given by the NMC, but until then we are going to focus on what we have right now. Okay, clear? So now end of this discussion, end of the syllabus, like क्या पढ़ना है हमें क्या नहीं पढ़ना है, whatever we have right now we will going to study that only. So today we will be starting with a chapter which is photosynthesis, and you know that this photosynthesis chapter is very pathetic, right? We have lots and lots of cycles here and you feel very bad you don't remember those cycles, Z scheme and all those things. So I make you a promise that after the finishing of this chapter, like after when we finish this chapter, you won't have a single confusion about this particular chapter, right? This is an introductory video, like introductory video for the chapter. So today we will going to learn about the basic photosynthesis, and then we are going to start about some of the things like experiments, scientists, and that's all. After that, in the next class we are going to go with the main basic terminology, main terms and cycles and all those things. Clear, everyone? Got it? Now let's start the session very quickly. So right now, first of all, in class 10th also, in class 7th also, in everywhere we have learnt about the photosynthesis. So if I ask you what is photosynthesis, so you should not be very blind, because we have already did this lots and lots of time in class 7, 8, 9, 10, everywhere, right? Sorry, right? Okay, so still I will be helping you from the basic. So first of all, the word photosynthesis is what? The word photosynthesis is made up of two words, as you can see, which is photo and the other one is the synthesis. So sir, what do we mean by photo? Not that photo, right? Not that photo which you click from your iPhone. No, not that photo. Photo here means what? Photo here means the light, right? Which kind of light, sir? The sunlight. And what does the synthesis means? Here the synthesis means what? Synthesis means to synthesize something, to make something. Yes? If you are good in English, your bio will be good, because half of the terminologies, if you are able to understand with the help of English language, you are going to crack that definition. I always tell my students that if your English is good, your biology is good, but that's just a thing. Hindi medium students also have lots and lots of knowledge for biology, but this is what I say to my students. Now, so synthesis means what? Synthesis means to make something, to make something. Clear? Great. Now sir, we know the meaning of the photosynthesis, like the word meaning we know. Now what, sir? What kind of process is it? So this process is what? Can I say that it is a physiochemical process? Yes or no? Yes, definitely. some of the physical things is happening, light is there, and some chemical things is also happening. So can I say it is a physiochemical process? Okay, why it is, how it is, when it is, who discovered this, everything we are going to discuss. But first of all, let us start with a step-by-step manner, right? Today only if you will ask me, sir, Z scheme, explain me the Z scheme, it's not possible. You need to know some of the basic things. So it is what? It is the physio. Now I have a very difficult in writing the spelling of physio. Physiochemical process. Yes or no? It is what kind of process? Physiochemical process. Now, from the starting childhood, when you started to love biology in class 10th and you decided that you will take biology in class 11th, and when you came in class 11th, your dreams got shattered that, oh no, biology is not very easy. From that time only, you know the definition, you know the equation for photosynthesis, right? So equation for photosynthesis is what? 6 CO2 plus 12 H2O, and this 12 H2O gives what? Gives glucose. What's the formula for glucose, guys? C6H12O6, and then what we do? We get 6 molecules of H2O, and then we get 6 molecules of O2. Yes or no? It is correct. I have written this correct. Okay. So we have 6 CO2 molecules and 6 H2O molecules. And then here glucose is made, and along with glucose, water and oxygen is also released. But this reaction should be happening in the presence of one thing, which is sunlight. Okay. When you are in class 10, if you won't write this sunlight, they will deduct your 1 marks. And then you have to write that in presence of chlorophyll. Okay, this two thing was very necessary, right? Okay, sir, but can you know, like, do you think that who was that first person who said that ki photosynthesis will only happen in presence of sunlight? Think about it. There might be some person, right? There might be some person who have said you that photosynthesis should happen in presence of sunlight, or it happens in presence of sunlight, or it happens in presence of chlorophyll, right? Something like this. So there were some very early experiments. Okay, so first of all, there was an experiment in which a person took a leaf. Okay, a person took a leaf. Very sure, sir. Let's draw a leaf, and my drawings are very good, you know, very good, very nice. Okay, now this leaf was covered with a black paper. Okay, covered with a black paper. So my background is black, so if I'll draw black paper, it will not show you, so manage with the blue paper. Okay, we have covered this with the black paper. That's very good, sir. Okay, that's very good. We have black paper. Now what, sir? If you have taken the black paper, apply your brain, sunlight is not going to penetrate that black paper. Exactly. And if sunlight is not going to penetrate that paper, so right now we know that photosynthesis was not taking place. But at that time, that scientist was very excited. He came and he opened that black paper and saw, oh my God, I discovered something. You know what I discovered? I discovered that photosynthesis doesn't take place in absence of sunlight. Clear to everyone? So this was the first experiment. This was the first experiment. Okay, clear. Now let's move on with the very basic experiment again, which was half leaf experiment. So half leaf experiment is also known as Moll's leaf experiment. Okay, it's the name of the scientist, Moll's leaf experiment. Clear? If you have, in case of you have any doubts, but you are like taking my session, so you won't have any doubts, but in case you have any doubts, so let me know in the comment section. So we have Moll's leaf experiment. Now in this experiment again, they did this thing, so someone has to do something else also, right? So what they did, they took another leaf, okay, a very beautiful leaf drawn by Bassi. This was the leaf. And now what they do is that they covered half part, the whole half part, and they kind of put that half part into a jar containing KOH. Okay, so I don't know how to make a jar, but I will try to make it. So suppose that this is a leaf. Let us experiment with some of the leaves. If they can experiment, we can also experiment, right? Oh my god, this is the leaf. Okay, wait, I will be drawing a very good leaf. So this is a leaf, and half of the leaf was put under a jar which was containing KOH. KOH. Now, you are making fun of my leaf. Now, if I'll ask what the role of KOH, then I'll make fun of you. Should I? No, because you must know this probably. So this KOH is what? This KOH basically, it is a carbon dioxide absorber. It absorbs carbon dioxide, okay? So it absorbs carbon dioxide. Now, recently, if you are with your full attention, recently we discussed that there is what? There is the necessity of carbon dioxide in photosynthesis. Yes, sir, very nice. There is that necessity of carbon dioxide in photosynthesis. If there is no KOH, if there is KOH, no carbon dioxide, and if there is no carbon dioxide, then how photosynthesis gonna be performed? So this two experiment proved that number one thing, sunlight is necessary. Second thing is that carbon dioxide is necessary for photosynthesis. Clear to everyone? Great. Right? So these were the two experiments which we discussed some of the basic part of the photosynthesis. Now, from here, questions can arise. In this two part is basically just for the reference in NCERT. It's given in a very short form in the first page. We need to discuss about this. But now this two part, which is Stephen Hale's important scientist, this is very much important, and this thing is asked in your NEET examination. Clear? Okay, sir. Great. Now what? Now this Stephen Hales is known as father of plant physiology. Stephen Hales is known as father of plant physiology. Why? Why do we call someone a father? Because he gave birth to something, exactly. So Stephen Hales was the first person to study about these things, and so we considered them as the father of plant physiology. Let's give him some respect. Father of plant physiology. I hope my video, my voice and my everything is clear to you all, right? Let me check. Should I check it once? I should take precautions, right? Once again. Father of plant physiology. Yes. That's very good. So, everything is going good. So, Stephen Hales was known as the father of plant physiology. But sir, we are Indians, right? We need to know about something which happened in India. So, father of Indian physiology is what? Father of Indian physiology is, can you guess? Father of Indian physiology. I will give you a hint. His name ends with Bose. His name ends with Bose. No, not Subhash Chandra Bose. How can he be the biologist? Huh? The name is J.C. Bose. The person name is J.C. Bose. Who is he? J.C. Bose. So J.C. Bose is known as the father of Indian plant physiology. Clear? Okay. Now next came what he did, what the father of plant physiology, which is Stephen Hales, did. So Stephen Hales said that there are two things which is responsible for photosynthesis. One is CO2 and the other one is the light. Clear? So he said two things is responsible for photosynthesis. One is CO2 and one is light. Don't laugh, okay? Now you all know that CO2 and light is present. At that time, he was the first person to discover this. So imagine the hard work he has made. For us now, to, what is this big? What big is in this thing? No, but it was very big, right? So CO2 and light. He said that CO2 and light is necessary for the photosynthesis. Clear to everyone? Yes, that's great. Now let's move on and now talk about the Joseph Priestley. let's move on and now talk about the Joseph Priestley. So he was a very chill pill guy, okay, and he was born around 1770. He was a dude at that time. Sorry, I made a mistake. 70 he performed the experiment. Okay, he was not born. He was born somewhere in 1733, I believe. So his experiment was done in 1770. So what he did, one day he was going around, okay, he was moving around like this in a farm, and he saw a mice, chuha, mice, right? He saw a mice and he saw that, okay, let's do some experiment. So he took the mice, put that mice on the table, and he was sitting and thinking that what experiment should I do with this mice? And then the mice was trying to run away. So what he did, he took a bell jar kind of thing and he put the mice inside. And then mice was frightened because there was no light. So he also light up a candle, right? So what he did, Joseph Priestley was moving in a farm, catched a mouse, put that mouse on the table, but the mouse was trying to run away. So to prevent that, Joseph Priestley bought a bell jar. He kept it like this, and then since it was very dark inside the bell jar, so he with the help of matchstick, he light up a candle. Clear? Okay. But after a few minutes, Joseph Priestley was like, oh, what happened? Candle was gone, mice was also gone. So mice had RIP, mice dead, candle bujh gaya, right? Okay, clear. So he started thinking that, what, I killed a mice, I am a murderer, how can it be, when I'll go to heaven, what I'm gonna say to God? He started thinking all those things. But then again he thought that, what if the same plant which is present in the farm, I keep that same plant in this bell jar? So maybe the mice can survive. And now very happily he again went to the farm. He again took a mice, and at that time he promised that mice, okay, I'm not gonna kill you. Again he did the same thing, and this time he also bought a plant. And now after few minutes, the Priestley was just observing the bell jar. Okay, till now it's 5 minutes, not died. 10 minutes, didn't die. 15 minutes, didn't died. 1 hour, didn't died. And he was very happy. So that means something was there in the plant, due to which the candle also didn't didn't gone, and this mice was also not dead. So candle was also completely fine, mice was also completely fine. So now Joseph Priestley was a very intelligent person, so that's why he had figured out that something is there which mice and candle is using, and the plant is again giving that thing into the environment, and that's why the mice and candle is now totally fine. Yes or no? Clear? So this thing was, in this thing, what he concluded, what things this Joseph Priestley did. So Joseph Priestley said that a burning candle, he said that, one second, a burning candle, or what, or an animal. So whenever you see a mice, you should be very respectful for those animals. Animal that breathe the air, that breathe the air, right, somehow damages it, somehow damages it, right, somehow damages it. But okay, now what kind of plants had he put? Mango tree, banana tree, papaya tree? Guys, how can these be put on bell jar experiment? He took a mint plant. The plant from which you make mojito, right? You crush the lemon and you keep that mint, and then you add some cold drinks and you drink and you say that, and then you post pictures on the Instagram. That mint plant only. But mint plant somehow did what? Somehow maintained the quality of air. Somehow maintained the quality of air, right? So this was all about the Joseph Priestley. This was all about the Joseph Priestley. So whoever is making the notes or want to make the notes, take a screenshot, or otherwise I'll ask my team to upload a drive link for this particular class. Okay. Now let's move on and let's talk about the another thing which says what? Which says Jan Ingenhouse, right? He was more intelligent than Joseph Priestley. He was like, okay, Priestley, you have done this. I am also a botanist. I can also do lots and lots of things. So this particular Ingenhouse was thinking, and then he was like, let's do something creative. He did from farm thing, I'll do something from water. Okay, so instead of mice, he was a kind of person that he doesn't wants to kill an animal. Ingenhouse was a kind of person that he doesn't wants to kill animals, very emotional person. Okay, so what he did is that he went and he took a plant. Okay, he went out and he took a plant, and that plant was also an aquatic plant. What kind of plant? That plant was an aquatic plant. Okay, so do we have to learn the name of that aquatic plant? Yes, sir. Definitely. So the aquatic plant was hydrilla. The aquatic plant was hydrilla. Okay, it's not visible. See now, the aquatic plant was hydrilla. Clear? Now what? He took that hydrilla and he put the put it into a beaker kind of thing, and then he took an inverted funnel and just placed it like this. Okay. Suppose that this is a glass. Do we have water in this? No. So he put this glass. Oh, wow, it's water. So we put that Priestley put the funnel kind of thing like this, and inside this, what he was having? He was having the hydrilla plant. Okay, great. Now who is going to clean this drop of water? Now, clear? Okay, sir. Now, this setup was kept in the sun, and when Ingenhouse was staring at this particular setup, he saw some bubbles. He saw what? He saw some bubbles. Now, at that time, he was not very much intelligent to say that this gas bubble belongs to oxygen, but right now to we say, so he said that some bubbles have came, right? Now, he also said that maybe if I do the same experiment at night time, maybe I'll be able to see the bubbles as well, right? So at night time, Ingenhouse was not sleeping at that time. He was not scrolling Instagram like you all guys do. He was not a person like that person. He was watching this particular plant. So he was not able to find a single drop of bubble. Clear? Okay. So at that time, this Priestley said that, okay, this thing was closed. Okay, this thing was closed. So Ingenhouse said that sunlight is necessary for the photosynthesis. So Priestley said, Ingenhouse said what? Sunlight is necessary. Sunlight is necessary for photosynthesis. So for photosynthesis. Now don't comment on my spellings, okay? The sunlight is necessary for photosynthesis. Clear, sir. Now, next person came whose name is J.V. Sachs, Julian von Sachs. Okay, This person was very kind of a person. He was very hardworking person. Before experiment, he used to carry sacks. Okay, he used to carry sacks, bora. He used to carry sacks, and he used to work in a sugar field. Okay, so now this person did the experiment on the what? He said that whatever the things which is made in the plant during the photosynthesis, that is glucose, and that glucose is stored in the form of starch. Clear to everyone? Okay, let's talk about the things what he did. Okay, so now he said that in photosynthesis, what thing is released? In photosynthesis, what is formed? Glucose is formed. Okay, which is C6H12O6. Right? Clear, sir. Now what? Now this glucose is formed from where? From stem, leaves, fruits? No, it is formed from green plants. It is formed from green plants. Clear? Great, sir. Now what? Now he said that this glucose is what? This glucose is stored in the form of starch. Okay. Now he also said one more thing, that if you are able to see the green color, this green color is due to a pigment which is present in a special bodies, right? This green color is what? It is due to a pigment which is present where? Which is present in a special bodies. Right now we know that these special bodies were what? These special bodies were you don't know? It is chloroplast. So at that time Sachs was not knowing it. Okay, so this was all about the Sachs. Now again Priestley came, that mice person. Okay, then we had this particular person which is engine house. After engine house we have this. Now you can see slowly the view of the study of photosynthesis is increasing. Now this Angel man was very intelligent. Okay, this Angel man was very intelligent. He was like, I'll do something innovative, and he did. He is a person who should be praised a lot. So what he did is that he took a prism, like you steal those prism from your physics lab in school, right? I know you guys do these things. So you have stolen that prism. Now when you go home, the Angel man also did the same thing, and from his school he stole a prism, and then he was like, this white light when passed through the stream, prism, it creates Vibgyor, right? Now he thought about that, what if I put a plant inside a water, and on that plant I'll throw this Vibgyor. So what things is gonna happen? And he observed that in the blue region, and in the green region, there were maximum droplets of oxygen. But he was also very intelligent. Like if I and you go and ask this Angel man, like, how can you confirm that this is the droplet of molecule of oxygen only? So he asked Azotobacter also. He placed Azotobacter, and Azotobacter was found in these regions only where the photosynthesis was maximum. And this person was the first one to discover the absorption spectrum of photosynthesis. This person was the first person to discover absorption spectrum of photosynthesis. Clear? Photosynthesis. So we can learn biology very easily with the help of these stories and all, right? It's like if you're gonna rote learn it, nothing is going to happen. If you build up a story in your mind and imagine that story, see how well you're going to remember things in biology. And it's my promise that I'll make your biology very easy. Now, okay sir, fine. Now let's go move on. Wait, I'm going. Next, it comes Neil. Okay, Neil was also a very nice person. He was more intelligent than Angelman. He said that, okay, you discussed that oxygen is released, blah, 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 and blah. So I will prove that from where this oxygen is released. So he said that if you take a green plant, so in green plants, H2O is the oxygen donor. So in the green plants, H2O is what? H2O is the oxygen donor. Clear to everyone? In the green plants, H2O is the oxygen donor. Clear? Great, sir. But there is some kind of bacteria which is what? Which doesn't like oxygen. They say that no, I won't do photosynthesis from water, bro, because I don't like you. So for them to do the photosynthesis, there is another thing, which is what? Which is H2S. Which is what? Which is H2S. Clear? So in green and sulfur bacteria, the photosynthesis is done by the help of H2S. So in this case, there will be no release of oxygen. Obviously, na, from here, if in this case, if I write oxygen, so you will get oxygen here, right? And if I write sulfur, so you will get sulfur here. Clearly, it is clear, right? Oh, sorry, sulfur here. It's very clear. So if there is no oxygen here, so how can you get oxygen gas? Right? So these gases, these bacteria, green and sulfur bacteria, are those kind of bacteria which performs the photosynthesis. They have everything, but they do not perform an oxygenic photosynthesis. They do not produce oxygen. So such kind of photosynthesis is known as anoxygenic photosynthesis. Okay, or you can say non-oxygenic photosynthesis. Right? Let's write non. An is kind of, sounding very weird. Non-oxygenic photosynthesis. Clear? Yes, sir. It's clear. We are very intelligent, sir. Everything is clear. But when we go to the exam, sir, what happens to us, we also don't know. Right? So green plants is what? If I talk about the green plants, so the green plants are performing those kind of photosynthesis in which oxygen is released, and in this, when the oxygen is released, such type of photosynthesis is known as what? It is known as oxygenic photo, where should I stand? Yeah, oxygenic photosynthesis. Clear to everyone? Right? Okay, should I move forward? Great. Now what, sir? Now comes Reuben and Kamin. They were what? They were very advanced. Reuben and Kamin. They were very advanced. So none of them used some kind of extra thing. Everyone was like, okay, whatever the thing are present, let's do it somehow, experiment. But Reuben and Kamin were extraordinary. They were very extraordinary. They said that, guys, I am going to use radioactive isotope. Okay, let's see what they have done. So they used what? O18 isotope. Now why my pen is not working? It's Reuben and Kamin, bro. You have to work. So O18 isotope is what? O18 isotope was radioactive. Okay. Now no one is going to ask me what is the isotope. Okay. Ask from your chemistry teacher. So this radioactive he used radioactive isotope in place of this. So let me write equation. So this was 6CO2 plus what? Plus we got 12 H2O. Now this oxygen is not normal. It is very high standard, just like Reuben and Kamin. So it is what? It is O18 isotope. Clear? Great. So see, can you see this? Only the hydrogen was having the O18 isotope. Now what? Now it went and the reaction continued and we get C6H12O6 plus 6H2O plus 6O2, and here we have the radioactive isotope. That means, which proves that whatever the oxygen we are getting, whatever the oxygen gas we are getting, it is coming from a very high standard oxygen, which is O18 isotope. So clear, Reuben and Kamin was a very high standard person. He used a very high standard thing, which is O18 isotope, and in this way they proved that the oxygen which was released from during the photosynthesis, it is released from the water, not from the carbon dioxide. Why? Because this, if it was released from this, so this oxygen should be normal, right? But it is having the O18 isotope. Clear to everyone? Great. Is it clear? Hi Tanish, how are you? Welcome back Tanish. Where were you from so many days? Now, let's have some questions. Tanish, my class will start from, oh my god, I have only forgotten. My class will start from 6:30 to 7:30 daily on this channel, okay? I want to see you in my class daily. Now, let's solve some of the questions. Which of them perform non-oxygenic photosynthesis? So just ignore this word photo because the space was not there and I have ignored it. So you also ignored. So which of them perform non-oxygenic photosynthesis? All alga, all plants, blue and purple bacteria? None. Tell me the answer in the comment section now. Tell me the answer in the comment section now, everyone. Okay, Sid is also here. That's very good. Yeah, the answer is what? The answer is option number C, blue and purple bacteria. Algae, oxygen. Plant, oxygen. Blue-green bacteria, sulfur, right? So this is why we are calling that this particular thing is what? This particular thing is non-oxygenic photosynthesis. Clear? Okay, sir. Now, next question. Who gave first action spectrum? Who was that person? Who was that intelligent person who thought that Priestley did this experiment? I will also do this experiment. Who was that person? Who was that person? Engine House? No, Engine House was not that person. It was an intelligent person, Engelmann. It was an intelligent person, Engelmann. Very good. Yes. It was an intelligent person, Engelmann. E for intelligent, you remember it like this, okay? Now, who is the father of Indian plant physiology? Very good, Tanish, very good. Who is the father of Indian plant physiology? Ramdeo Mishra, Subhash Chandra Bose, Jagdish Chandra Bose, and Priestley. Subhash Chandra Bose, Jagdish Chandra Bose, and Priestley. I'm just laughing because someone can also choose wrong answer, right? Okay, the answer is what? The answer is C, which is Jagdish Chandra Bose, not Subhash Chandra Bose. Okay, Ramdeo Mishra is some related to ecology. We'll read about that thing in ecology. And Priestley, it's not an Indian name also, right? So Indian parents can't name their child Priestley. They will only not able to pronounce the name. So how can they name these kind of fancy names, right? So Priestley ko to aise hi hata do. So we have these three options and the main option is Jagdish Chandra Bose. Okay, that's very good, Tanish. Now, which among the following light performs maximum photosynthesis? ये थोड़ा सा grammatical mistake हो गया यहाँ पे. So in which of the following, समझ लो ना क्वेश्चन को, in which of the following maximum photosynthesis occurs? Red light, purple light, green light, or yellow light? We have red light, purple light, green light, or yellow light. So action spectrum intelligent person, action spectrum intelligent person, the person who stole prism from his school, Engelmann, exactly. And Engelmann said that in red light or in blue light maximum photosynthesis takes place. Yes, sir. Tanish, we are going with the photosynthesis as of now, but I'll try to start genetics also again. Okay, we will complete photosynthesis and genetics together. I'll update the schedule. Okay, so here we have red light, here we have red light. In red light we are going to, in red light maximum photosynthesis is going to happen. Is it clear to everyone? So I hope that you like this class, and for the notes what you have to do is that I'll ask my team to upload this notes through a drive link or something, whatever they can do. I'll ask them. And remember that I am planning to take 2 classes per day, which will be from 4 p.m. to 5 p.m. Okay, which will be for 4 p.m. to 5 p.m. In this, I will be trying to complete your class 11th portion. And in this time, which is actually from 6:30 to 7:30, but today there was some problem with my laptop and I came late. So 6:30 to 7:30, I am planning to start with 12th. So if you have like comfortable with this time, please do come in my class, because if you're gonna watch live lectures, you will learn little more, you will get motivation and all those things. But if you're not able to come live, watch the recordings and share the feedback of my class. So I hope that we are going to complete this particular biology syllabus till December. I'll try my best to complete the biology syllabus till December, and from January, February, March we will revise it, and then we will have mock tests and all those things. Okay, so stay tuned for more updates. This channel is going to bring lots and lots of things for you, everyone. Till then, bye bye everyone. Do visit my class at 4 o'clock and 6:30 p.m. Bye bye everyone. See you.